Hello, welcome to the screencast where we're going to address the gross anatomy of the eye. And here we're going to look to identify and describe the three tunics and their structures and functions, as well as describing and identifying gross anatomical features of the eye. So firstly, the three tunics are the fibrous outer tunic. The second tunic is going to be the vascular tunic, and the third one is the sensory tunic. Right. Now, a tunic, for those of you that don't know, is kind of like a cloak, right? A jacket, and so these are just layers of the eye. And again, fibrous, vascular, and sensory. So if we look at the sagittal suction through the eye, let's first identify the structures involved with the fibrous tunic. So the sclera and the cornea are the two structures that are part of the fibrous tunic. Posteriorly, the sclera is opaque and white, and this forms the white of the eye. As you move more anteriorly, the most anterior portion of that becomes clear and colorless, and is what we call the cornea. Right? So this is clear and colorless. Right. So that cornea is clear and colorless, will allow light to pass through it. The sclera, opaque and white, and is tough and gives the eye protection and this round spherical structure to it. The next tunic underneath that is the choroid layer here, the choroid tunic, and this is the vascular. And anything that's vascular means it has blood vessels going to it. So if you look at the cartoon here, you can see the blood vessels here in the choroid. These blood vessels originate from this optic nerve, and as it comes in the back of the eye this way. So blood vessels will be part of that optic nerve there, cranial nerve number 2. More on that later. So this choroid layer posteriorly and laterally has lots of blood vessels serving the cells of the eye. As you move this vascular tunic, this choroid layer, more anteriorly, it's going to split and divide into two structures. The more anterior of those structures is called the iris, and this is a pigmented or colored muscle. And then posteriorly, that choroid layer will split and divide and become the ciliary body, which is also a muscle. So coming down from the ciliary body are the ciliary zonule. Right? The ciliary zonule here is a ring structure that goes all the way around the lens, which we'll talk about in just a second. But the ciliary zonule are like tendon-like structures that will, as you see here, support the lens. So they're attached to the lens and they're attached to the ciliary body. So the lens is a very clear protein structure that has the ability to become more convex, more concave, and so it will change its thickness. And so sometimes it's really stretched out and thin like this. Other times it will become more convex like this. And that the job of that lens is to focus light as it comes through the eye. So by changing its, whether it's stretched out like this or whether it's more convex like that, concave on either side, you're able to focus light to the back of the eye. So that brings us, while we're up here at this anterior end of the eye, let's say that this lens right here is actually separating two chambers of the eye. The anterior compartment here contains fluid, and the posterior compartment, or the posterior segment, contains a fluid as well. Let's talk about the anterior one right now. The fluid that's in there is called aqueous humor, and it is very watery, right? And it is produced by specialized cells in this around the ciliary body, right down here, and also up here. But that aqueous humor is produced, and it will move between the lens and come out the opening right here of the iris, right? And will be circulated around the eye, and then it's going to drain out of little canals here called the scleral. Right? This is the sclera, right? venous, right? It's a passageway, and a sinus is a cavity. So this little hole is the scleral venous sinus, also known as the canal of Schlemm, one of the best last names in all of biology. So that aqueous humor, again, produced by specialized cells in the ciliary bodies, and it's constantly produced and circulated there around. While we're here, there's the iris again. This is the pigmented colored part of the eye, and it's not continuous, right? You don't have the muscle doesn't continue right there. There's an opening or a gap, the pupil. So the pupil technically is not a structure. This pupil is a gap, and that's where light will actually enter the back, the posterior segment of the eye, or that's how it gets into the posterior segment of the eye, is through that pupil there. So let's talk about that posterior segment. That is filled with another fluid. It's called the vitreous humor, not aqueous, but the vitreous humor is gelatinous. It is really jelly-like, okay? And so the vitreous humor here provides some pressure, and it keeps helps keep the eye with it, giving it the eyeball its shape, as well as with this sclera. Giving it that round shape there. But this vitreous humor is gelatinous, and it's much more solid than the aqueous humor anteriorly. And this helps to also keep the third tunic, which is the retina, and again, this is the sensory tunic, helps keep that retina up against the back of the eye here. So this retina is what is all in yellow here. So anteriorly, you see that the retina stops up here right around where these ciliary zonule are going to be. But posteriorly and laterally, the retina is full of sensory cells, right? And these are neurons and modified neurons, right? The rods and the cones, and we'll deal with that detail in another screencast. Right? So these are where the cells are that are going to be detecting the light. When they detect the light, they absorb the light. They will then send action potentials down and out the back of the eye, down the optic nerve. So this is going to go to the optic chiasma at the base of the brain, and then back into the optic cortex part in the occipital lobe. Two more points here, two more things to point out. One, in the retina there's this fovea. Fovea is a pit, right, or a little depression, and it's central. And so this pit or fovea centralis, which again we'll deal with more when we look at the sensory tunic here, this is where light waves are going to be focused. So as light waves come in through the cornea, in through the aqueous humor, they pass through the lens, and the lens will focus those light waves, bam, right there on that fovea centralis. Right? So this is kind of important. That's the fovea. And then the last structure that you see at the back of the eye is this thing right here, which is called the optic disc, which is also where there are this is where the blood vessels are coming in, and so there's no photoreceptors here. Right? So no photoreceptors. So there's no photoreceptors. This is also what's known as the blind spot, right? Where light waves, even if light waves are focused right there, there's no sensory cells to detect them, right? And so you don't send action potentials down the optic nerve here, and you don't register anything. And we'll do a little demonstration in class to show you how you can see when you're focusing light waves on that blind spot. Okay? So lots of structures there, lots of detail in the eye to take a look at. We give you a blank one to sort of practice labeling this with, sketching this on your own, drawing it, labeling it. Is probably the best way to kind of get this all down. Let me show you one more slide here that deals with, again, a section through the eye. So here's the sclera, right? Anteriorly, here's the cornea, here's the aqueous humor. Here is the choroid layer, which becomes the iris anteriorly, and posteriorly becomes the ciliary body. Here are your ciliary zonule attached to your lens. Right? So when you look anteriorly at the eye here is the lens, and here are the ciliary zonule, right, that then become continuous with the rest of that choroid layer. Right? So you'd have to really peel the eye back in order to see this. And we'll do the dissection, and you can see that ciliary body. The other image on this slide here is when showing you the pupil. And again, remember the pupil is not really a structure, it's just actually a gap there where light waves are going to come in through the eye. So it's surrounded though by the iris, which actually has two layers of muscle in them. And so this inner layer right here is circular, and when that muscular layer contracts, the pupil gets smaller, and we say that the pupil constricts. The other layer of muscle is outside of that, and the fibers run more radially, right, instead of circularly, and this is causing the pupil then to dilate. So what is causing this? Well, what's causing this dilation or constriction is, of course, cranial nerve number 3, which is oculomotor. But now remember that oculomotor has parasympathetic and sympathetic stimulation to it. So which one would cause constriction? Which one would cause dilation? If you said parasympathetic causes constriction and sympathetic causes dilation, you are correct. So when we sympathetically stimulate the eye oculomotor, we send sympathetic pathway down, you're going to contract these radial ones, but then the circular ones will relax. And conversely, when you stimulate parasympathetically, the circular ones contract and the radial ones will relax. And this is how you control the how much light is coming in to your eye. Right? So, sure, lots of information there to practice with. We will dissect an eye in class. This is a real eye, sagittally sectioned, and we'll try to take a look at that in class at some point. You have some blanks to practice labeling, and you have some coloring book pages as well. So have at it and bring your questions, confusions to class. Thanks.